Welcome everyone to this presentation about antibody binding kinetics measured by the newest tools of the switchSENSE technology. I'm Nena Matscheko and I'm heading the Cells and Antibodies Research and Development team at Dynamic Biosensors. Dynamic Biosensors has developed the proprietary switchSENSE technology. For everyone who has not heard about switchSENSE before, this is technology specialized in interaction analysis. The analysis of on and off rates as well as conformational changes positions the technology in the preclinical drug development. The high information content and quality of data is especially well suited for the lead identification and optimization. Our newest developments, which you will see during this presentation, are now spanning into in vitro cellular assays, providing even more binding and specificity information on drug candidates before entering the more expensive in vivo studies in animals and humans. During this presentation, I would like to introduce you to two topics. I will start with familiarizing you with the switchSENSE technology, in which you immobilize a purified binding partner on the surface and then you use the second binding partner in solution. I will show you examples of otherwise difficult to access measurables, especially from the field of multispecific antibodies. This will include avidity effects, dependency on steric orientation, and on ligand density. Afterwards, I will give you an exclusive insight into the recent developments going on at Dynamic Biosensors. This will be especially interesting for you if you think it is advantageous to measure binding to membrane proteins in their native surrounding in the cell membrane. So let's get started with switchSENSE for multivalent binders. There are several technologies on the market that offer kinetic analysis. So some of them are focused on binding, affinity. Others are able to resolve the association and dissociation rates separately. So why use switchSENSE for your antibodies? This technology is especially suited for resolving not only a one-to-one affinity, but also a one-to-two or more avidity. It was never more easy to accurately and reproducibly control the density of the immobilized binding partner on the surface. On top of that, you can control the ratio of several ligands that you would like to measure at the same time. And when working with high affinity binders, you will need a stable measurement over long times, resolving dissociation times of maybe several hours. When measuring binding affinities of antibodies, most of you will be familiar with the immobilization of the antibody on the sensing surface and then injecting the antigen solution. But in that assay orientation, you cannot measure a cooperative effect of two binding sites, no matter if we're talking about two equal or two different antigens. In contrast, in switchSENSE, we are immobilizing the antigen on the surface and use the antibody as analyte in solution. This allows us to measure the individual interaction rates K on and K off of each arm of the antibody to its respective antigen. In addition, the antibody can also bind with both arms simultaneously, and we can measure the effective K on and K off of this cooperative interaction, resulting in the avidity dissociation constant Kd. The immobilization of antigens on a switchSENSE surface is achieved by a DNA hybridization on the measurement electrode, a very flexible and mild method for the ligand. The heliX biochip also features a second real-time reference spot, which can be used to detect non-specific interactions of the analyte solution with the surface. The data I'm going to share with you on the following slides was achieved with immobilizing the antigens as ligands on the surface and flushing a multivalent analytes, the antibodies, over the sensor. By varying the antigen on the surface, we are able to resolve both affinity as well as avidity of a binder. In this example, you can first see the recorded on and off rates of a monovalent Fab fragment against its antigen, which resulted in a 2-digit nanomolar affinity. When adding a second antigen on the surface and measuring the interaction of a bivalent dual Fab construct, the dissociation data revealed a significant portion of analyte being bound in an avid way, meaning with both arms at the same time. As you can see, the dissociation had to be measured for about 5 hours to resolve this slow dissociation process. The off rate slowed down by a factor of 65, resulting in a now picomolar binding affinity. It is easy to understand that binding by two arms is stronger than by one arm. However, it is not that easy to understand how much stronger this bivalent interaction will be. Unfortunately, we cannot apply the avidity effect of one sample set to another. This effect is influenced by many factors, including the individual binding affinities of each arm, as well as the steric orientation and flexibility of the construct. To illustrate this further, I would like to show you in comparison the rates of the therapeutic bispecific antibody Emicizumab, known by its trade name Hemlibra. It is replacing a dysfunctional factor 8A in the blood coagulation cascade that should bring the factors 9A and 10 into close proximity. It's a great application of a bispecific antibody. This antibody has very different individual binding affinities. Where we were talking about low nanomolar to picomolar affinities before, we are now facing individual arms of just micromolar to middle nanomolar affinities. The rate analysis shows that in this case, the avidity effect is very different. While the weak micromolar binder is still profiting by a factor of 15 times decrease for the off rate, the stronger binder can actually not profit at all from the very weak binding body. This illustrates that avidity effects are very difficult to estimate and calculate, and actually always need to be measured by determining the dissociation rate of the avid-bound antibody. The avidity of bispecific antibodies are gaining increasing importance with the development of new bispecific therapeutics. While many bispecifics are designed to connect two target cells, others are meant to bind to one target surface. The combination of two binding sites on one target surface is, for example, employed for increasing tissue specificity. In this literature example from a publication from Meninune, a Th17 target cell combines the surface markers CD70 and CD4. However, these markers are not unique to this cell type. CD4 is also expressed on Th1 cells and CD70 on B cells. If the employed bispecific antibody now has high individual affinities for each of these binding sites, we will see high off-target binding and therefore cell cytotoxicity to Th1 and to B cells. It is therefore advantageous to aim at low individual affinities, but a high avidity effect if both markers are expressed. This has been achieved here by mutating the CD4 binding site and lowering the individual affinity from 0.9 to 70 nanomolar. While the cytotoxicity of the target cells remained almost the same, the killing of the Th1 cells was significantly reduced. switchSENSE is a convenient solution for measuring these individual but also cooperative binding strengths. To measure the cooperative binding to two antigens and the avidity effect on the dissociation, we have to immobilize both antigens at the same time on our surface. The DNA-based switchSENSE chip allows full flexibility and control concerning the immobilization of antigens. If you are working with two different antigens, you can color code them by hybridizing one of them to red labeled DNA and the other one to a green labeled DNA. These two ligand strands are then mixed either at a 1:1 or at any desired ratio. Since the anchor DNA on the heliX biochip is the same for both colors, the ratio will be transferred to the chip when your ligand solution is hybridizing. You will end up with a ligand surface containing both antigens at your desired ratio. Then you can measure your individual affinities in the respective color. So the ligand, the affinity for ligand 1 in red and the affinity for ligand 2 in green. And then you can also measure the avidity effect, which will show up in both colors simultaneously. On top of the individual ratio of two antigens, you can also precisely adjust the overall density and therefore the distance of the antigens from one another. By saturating some of the anchor DNA strands on the surface with DNA only, so without ligand, you can easily create a low density surface. Let me show you now a practical example from our lab where we took advantage of the color-coded immobilization of two antigens on the surface combined with the density variation. A clinically targeted antigen pair is the combination of EGFR and HER2, since high expression levels of both of these receptors are connected to a poor clinical outcome for breast cancer patients. However, EGFR is a broadly expressed receptor in many tissues. It is therefore important to develop an antibody which relies on an avidity effect with HER2 for its specificity. While the off rate from EGFR alone of this antibody was rather fast in the 10 to the power of -3 range, the residence time increased about 100-fold when the antibody was bound to HER2 as well. In vivo, the expression of HER2 is generally lower than EGFR. Therefore, the effective off rate depends on the density distribution of these two antigens. We could mimic this with different ratios of HER2 being immobilized in relation to EGFR. As you can see in the plot on the right, if only a very weak expression of HER2 is mimicked, below 50% of the antibodies can bind in a cooperative manner. The ratio of avidity and therefore the slow dissociation doubled to more than 80% when 1/3 of the immobilized receptors was HER2. This leads to a longer residence time on target cells and lowers the off-target binding. In summary, therefore, it leads to increased safety of the bispecific antibody. The easy control of the ligand density on the switchSENSE surface, as well as the adjustable ratio of several antigens, allows to mimic target and off-target cell surfaces in the heliX biosensor. Yet, we also know that membrane protein targets might change their properties when undergoing a purification process. Considering that 60% of all antigens targeted by current antibody therapeutics actually are transmembrane proteins, it would be desirable to measure kinetic rates not only on purified antigens, but actually on antigens in the native surrounding in the cell membrane. This will ensure correct folding of the membrane protein. The cell will directly display the antigens at the relevant density, and possible effects of the antigen-antibody interaction due to neighboring proteins can be taken into account. For this reason, lead compounds are always tested in cell culture before entering in vivo assays. However, these assays normally do not include kinetic rates measurements, but indirect readouts such as cytotoxicity. This is mainly due to a lack of commercial technologies for the determination of kinetic rates on cells. The standard method for measuring binding of antibodies to cells is flow cytometry. FACS is a commonly used method to identify if a certain marker is expressed, or if an antibody is binding to how many cells of the population. However, this technology cannot determine on and off rates, because the method is not based on surface immobilization of one binding partner. Even KD values are tedious to achieve. So despite being a good method for a quick check if binding is taking place, another method needs to be applied for understanding the kinetic binding behavior of antibodies. The currently most common method for measuring kinetic rates are SPR systems. They have recently even been equipped with an imaging system, which allow the identification of cells settling on the sensor and the observation of morphological changes. Yet the actual purpose of SPR, so measuring the kinetic rates by flowing buffer over the sensor, is difficult to combine with cells, since the buffer will easily wash away cells when buffer flow is applied. Measurements have only been performed at a very low flow rate, which generally leads to mass transport limitation and the rebinding of analyte. Another limitation of the surface plasmon resonance sensor is the limited detection depth of about 200 nanometer, which means that cells, which are in a micrometer range size, will only be measured at the very bottom where they sit on top of the sensor, but not on the exposed surface on top. This leaves a gap in the market for a commercial automated system that can measure kinetic rates of analytes binding to cell surface markers. We at Dynamic Biosensors are therefore working on immobilizing entire cells as ligands on our biosensor. This has been made possible now with the new heliX biosensor. To achieve stable immobilization of cells within the microfluidic system, we have developed a label-free immobilization technique for all cell types. We take advantage of the multi-way microfluidic system of the heliX for resolving rates without measurement artifacts, such as rebinding. The heliX is equipped with both light microscopy detection as well as a dual color fluorescence photon counter, which allows full visual control of the cells and sensitive interaction detection on the entire cell. And finally, you do not need to stay away from cell culture media or any complex solution, since this will not interfere with our sensing method. These features, together with the specialized software for automated data analysis, now allow the real-time measurement of kinetic rates on cells. On the following slides, I would like to explain in more detail how the cell immobilization works and show you some first data from our research lab. So how do we use entire cells as ligands in a biosensor? Up here you can see a heliX measurement chip equipped with 2 simultaneously observed measurement spots. Based on the standard heliX chip, we have developed special cell trap chips, which instead of anchor DNA carry a trap for individual cells on the measurement spot. While spot 1 is functionalized with the traps, the spot 2 can serve as a reference spot for background signals. The automated microfluidic system can transport cells and antibodies from the sample tray of the heliX to the chip, where cells get trapped and analytes can be observed by a fluorescent tag on them. These traps are physical barriers in the microfluidic system and are therefore not dependent on any cell-specific anchoring molecule. This means that they can be employed for all cell types, both adherent as well as suspension cells. As you can see on this same micrograph, the traps are designed in a football goal-like mesh structure to allow unobstructed fluid flow and therefore optimal transport of the analyte to and from the immobilized cell. These traps are produced by 2-photon polymerization and are very stable in even high flow rates. Let's now take a look at the traps as seen in the bright field microscope of the heliX during cell capturing. The cell solution of about 100,000 to 1 million cells per milliliter was placed in a temperature-controlled autosampler of the heliX device and then taken up automatically and transported to the measurement spot. The visual control of the heliX allowed to observe the loading of the cell traps. All 3 single-cell traps on the electrode were filled within a minute. When you take a close look at this image, you can even judge the health of the cells, if they are still round and intact or already disintegrating and dying. This automated capturing of cells prepared the heliX for any subsequent kinetics measurements on top of the cell. The most important parameter for judging efficacy and safety of an antibody drug is the residence time. It is therefore especially crucial to measure the dissociation rate of a potential therapeutic from the cell surface. I'm going to show you now one example of an anti-CD3 antibody interacting with Jurkat cells. Jurkat cells are immortalized T lymphocytes which show a high expression level of the CD3 antigen. CD3 is an integral part of the T cell receptor. The high expression level provides the opportunity of bivalent binding to the antibodies. The employed workflow to access the dissociation rate included the pre-incubation of the target cells with a fluorescently labeled antibody, comparable to the preparation of a FACS experiment. After sufficient incubation time, the labeled cells were washed and placed into the heliX device. The instrument automatically took up the cells and injected them onto the cell trap chip, where they got immobilized. To measure the dissociation in real time, the cells were continuously exposed to buffer flow while the fluorescence of the labeled antibodies was observed. Whenever an antibody dissociated and was washed away by the microfluidic flow, we could observe a decrease in the fluorescence. And these are the measurement results from this workflow shown before. So after pre-incubation of the cells with the fluorescently labeled anti-CD3 IgG, the cells were injected onto the cell trap chip. As you can see in the fluorescent trace here on the right, the capture of the labeled cells can be observed by a steep increase of fluorescence in the beginning of the measurement. The cell suspension was then chased with PBS buffer, and the dissociation of anti-CD3 antibody from stably captured cells was observed by a slow decrease of fluorescence over 20 minutes. This experiment had been controlled for autofluorescence of the cells, as well as the washout time of the fluorescent solution from the biosensor, ensuring that really only actual dissociation of labeled antibody from cells is leading to the observed slow decrease of recorded fluorescence. Within 20 minutes, we can observe a small fraction of antibody unbinding from the Jurkat cells with an off rate of about 2 × 10⁻⁴, suggesting a rather strong bivalent binding mode of this antibody. This type of measurement can be performed on various cell types, for example, to estimate resonance times of antibodies in different tissues. This will provide early on in development important information on both target and off-target binding efficiency, and is therefore closing the gap between in vitro and in vivo experiments. So with this recent data from our research lab, I would like to come to an end of my presentation now. I hope the variable surface functionalizations of switchSENSE have triggered your interest. And if you have any questions or you would like to try out the technology for yourself, please do not hesitate to check our website and get in touch with us. We are currently especially open to collaborations in the field of cell-based measurements. Thanks everyone for listening and enjoy the rest of the conference.